In this video, you're going to learn how to cross traits that are two different genes at the same time. So up until this point, we've only been doing one trait at a time. What color the peas are, what color their flowers are, or how tall a plant is. But now, you should be able to cross two traits at the same time. So will a plant be tall with purple flowers, tall with white flowers, short with purple flowers, or short with white flowers? So this is a two-factor cross. These are the steps to set this up. First, you're going to choose letters to represent the genes. I'm going to use some examples with these mice. In mice, the ability to run normally is a dominant trait. So mice with this trait are called running mice. The recessive trait causes mice to run in circles. That would be called waltzing. The dominant trait we represent with a capital letter, and the recessive trait is represented with a lowercase letter. We're also going to refer to hair color. Hair color, in this case black, is dominant, so we'll use a big B, and recessive is brown, so we'll use a little b. I recommend when you are doing crosses that you always choose letters in which the capital letter is a different shape than the lowercase letter. When you're typing, it's not as difficult to tell, but oftentimes you might be doing this handwritten, and it's difficult to tell the difference between a capital S and a lowercase s because they're the same shape. But if you choose a letter like R or B or H or T, it's easier to tell which ones are capital and which ones are lowercase. Once you've determined your letters, which we had already done here, you then need to determine the genotypes of the parents. So most of the time a problem will give you what the organism, the genotype is in words, and you have to figure out what it is in letters. So if an organism is heterozygous, that means that one letter is capital and one letter is lowercase. So it's heterozygous running, means that it has one big R and a little r. If it's heterozygous black, that's big B, little b. So this is just one parent. It tells us that it's running and that it's black and that it is heterozygous for both traits. The other organism is also heterozygous for running, so big R, little r, and heterozygous black, big B, little b. These are the parental genotypes. These are the genes that the parents have, and therefore the only letters that they can pass down to their offspring. If you get confused with your letters, sometimes I like to set up a little table here. Dominant versus recessive. You can set up a little table so we can see like its movement. Dominant is running and recessive is waltzing. And then next would be color. Dominant is black and recessive is brown. That way, if you get confused later, you can look back on that table and it'll let you know what your traits are. Okay, so once you have determined the parent genotypes, which we have already determined are big R, big B, little b, crossed with big R, big B, little b. Now we have to determine the possible gametes. So remember back from Biology 1, a gamete is a sex cell. This is where they're making the sperm and the egg. And this only contains half of the genes. So there's always going to be one R and one B in each gamete. But we have to figure out all the possible combinations in which gametes can be produced, because it's not always going to be the same R and the same B that are passed down. So we can use, sometimes you can just look at it and it's easy to tell, but if not, you can use the FOIL method. So the F stands for first. The first R and the first B. So one possible gamete is big R, big B. That's one. Then O, so first. O stands for outside. So the R on the outside and the B on the outside, which would be big R, little b. I stands for inside. So that would be the R on the inside and the B on the inside, little r, big B. And then L stands for last. The last R and the last B. Little r, little b. So now, these are the 4 possible gametes that this parent can produce. We'll do the same thing for the other parent here. The first R and the first B, same. The outside R and outside B. Inside R and inside B. Last R, last B. Then we're going to put the gametes on the top and the sides of our square. So it would have big R, big B was one of my gametes. Big R, little b was another one. Little r, big B, and little r, little b. That was one parent, so all of those gametes go along the side. Then the next one is big R, big B. This is the other parent. Big R, little b, little r, big B, little r, little b. So those are all the possible gametes that each parent can produce that go along the sides and the top. Then we just fill in the squares just like you would for a smaller Punnett square. So we'll bring down this R to here and this R here. There you go. So you should put the R's together and the B's together. And it's easier if you put the capital 1 in front so that it's easier to tell what the phenotype of the organism will be. But it's really important to put the R's together and the B's together like that. And R's and B's. And R's and B's. And fill in all of the boxes until you have something that looks like that. Once you've filled these in, then you can determine the phenotypes of the offspring. So phenotype, remember, is what it looks like. I'm going to use a highlighter to show what all of these are going to look like. Remember, looking back at your table, the big R stands for running, and the little r stands for waltzing. So any that have at least one big R are going to be running mice. They're going to run normally. Big B, remember, stands for black, and little b was the recessive brown. So now I want to count up how many of the mice are going to be running and black. If they have at least one big R and one big B, they're going to be running and black. So that would be this one, this one, this one, this one, this one, this one. Not this one here, because this one has 2 little b's. This one, and this one. So out of all of my 16 here, we have 9 that are running and black. Let's see how many are running and brown. So they have at least one big R and only little b's. That would be 1, 2, 3. How many are going to be waltzing? Sorry, waltzing is the little r's, but still black. 1, 2, and 3. And then of all of these 16, there's only going to be just one that shows both recessive traits. Little r, little b, little b. Sorry, let me choose. Okay. So now we can determine how likely it is for one of their offspring to be running and black. 9 out of 16, because 9 of the boxes out of the 16 total are running in black. Now we have to look back on the problem to see what question we're answering. So our problem was, if we have 2, how many of them are, or what is our ratio? That's usually going to be our question here. So our ratio this time would be 9 to 3 to 3 to 1. 9 running black to 3 running brown to 3 waltzing black to 1 waltzing brown. Anytime you have 2 organisms that are heterozygous for 2 traits, your ratio will be 9, 3, 3, 1. Go ahead and complete the practice for the dihybrid crosses and the two trait crosses. And please contact me with any questions that you might have in order to clarify anything that we did in this lecture today.